So we will now discuss about the next mechanism of eukaryotic gene regulation, that is your control of RNA stability. Now, one of the most important role that control that RNA stability is your UTR. UTR means untranslated regions. Now, what is actually RNA stability and how it is maintained? We have to see first. So, it is saying that before the mRNA leaves the nucleus, it is given protective caps, the 7-methyl guanosine triphosphate cap at the 5' end, which is catalyzed by the 3 enzymes, as you know. Okay, so before mRNA leaves the nucleus, it is given protective caps that prevent the end of strand from getting degraded during its journey, right? Now this cap, which is placed on the 5' end of the mRNA, is usually composed of a methylated guanosine triphosphate molecule, right? With the help of a GTP molecule by a nucleophilic attack, it binds, okay, and is catalyzed by 3 enzymes. Next, a poly A tail also, which is attached to the 3' end, is usually composed of a series of adenine nucleotides. Once the RNA is transported to the cytoplasm, the length of time that RNA residues there can be controlled. How much time that RNA will be remaining in that cytoplasm, that can be determined by some proteins, by some regulatory sequence, right? And one of those is UTR, the untranslated region. Each RNA molecule has a defined lifespan, and that RNA will be decay at specific rate, because if RNA remains permanent in the cytoplasm, it means all the time translation will be occurring and that protein products will be formed. This we have to regulate, we have to degrade that RNA after a particular period of time. In future we will also, in future if that protein is required, we will again go for the transcription and translation for that particular segment of gene. Okay, now next page. Each RNA molecule, whether it is mRNA, whether it is rRNA or tRNA, have a defined lifespan and they decay at specific rate. This rate of decay can influence that how much protein is in the cell. Okay, this rate of decay in the mRNA, wait for a minute. So this rate of decay can influence that how much protein is in the cell. If the decay rate is increased, suppose if you want to increase the rate of RNA degradation, then RNA will not exist in the cytoplasm for a long time. Thus, the rate of translation of that mRNA will be slowed down. So that can be used as a method of choice for inhibiting the translation of some pathogenic bacteria by decreasing the rate of transcription, by degrading their RNA, right? It can be used further for further in medical, right, in medicine research. So the rate of decay of RNA molecule will influence that how much protein will be produced inside the cell, in the cytoplasm more specifically. If the decay rate is increased, then RNA will not exist in the cytoplasm for a long time. It will not remain in the cytoplasm for a long time. RNA will not exist in the cytoplasm for a long time, thus the rate of translation of that mRNA will be low. Conversely, if the rate of decay is decreased, reversely, if you decrease the rate of decay, then RNA will remain in the cytoplasm for a longer duration of time and more protein can be translated. This rate of decay of RNA, whether it is mRNA or rRNA or tRNA, is known as RNA stability. So RNA stability means the time span, the lifespan of that RNA molecule, how much time it will reside in that cytoplasm and will get degraded after particular time. Binding of proteins to the RNA can influence its stability. Binding of protein to the RNA can influence its stability. If some regulatory proteins bind to the RNA, it can influence their stability. It can influence the stability of that RNA. There are some proteins known as RNA binding proteins. These RNA binding proteins bind to the region of RNA just up in the upstream or in the downstream of the protein coding region. Binding of proteins to the RNA can influence its stability. It can alter its stability. Maybe some protein will increase the stability of that RNA, or some proteins may decrease the stability of that RNA. So there are some proteins called RNA binding proteins which bind to the regions of RNA in the upstream or in the downstream of the protein coding region and influence their stability and influence its RNA stability. So these RNA binding protein actually, they don't bind to the codon region, they don't bind to the open reading frame region, but it binds to such regions other than open reading frames, which are known as untranslated regions, UTR. Now, what are these UTR? As the name suggests, untranslated regions. These regions don't get translated normally. Untranslated regions are the regions which are found outside the open reading frame. If you say in simple words, there is a more critical definition for untranslated region, but if you say in simple words, untranslated region are located outside the open reading frame, where your RNA binding proteins will bind and influence the stability of that RNA. Better to say, it regulates the mRNA. Actually, it regulates the translation or transcription process, post transcription and modification process, as well as translation process. So these UTR or untranslated regions are found just before the protein coding region begins, and it is also found just after the protein coding regions finishes. Means these UTR or untranslated region are found on the 5' side, known as 5' UTR, and it is also found in the 3' side, known as 3' UTR. So here is a little bit I have, for simplicity, I have drawn the picture, the 5' UTR and the 3' UTR. If you can able to see it very clearly, okay, you can able to see it now maybe. So this is the cap region, the 5' methyl guanosine triphosphate cap, 7 methyl guanosine triphosphate cap. Then comes your the upstream of the open reading frame, there comes the 5' UTR. Within this 5' UTR, there are many essential elements that you already know. Okay, that you already know, but now you cannot able to understand this, because until I explain about the 5 prime UTR, then you can not able to understand this. Okay, so what are the sequence maybe that can be found on this 5 prime UTR? If you guess, I give you time for this. Now this 5 prime UTR is found on the upstream, right? It is found just before the start codon of the open reading frame. And then comes the 3 prime UTR. 3 prime UTR is found in the downstream maybe, or you can say just before the stop codon of the open reading frame. You remember, open reading frame always begins with a start codon and ends with a stop codon. So 5 prime UTR found before the start codon, and 3 prime UTR found just after the stop codon. So both 5 prime and 3 prime UTR are found outside the open reading frame. Okay, then it is followed by a poly A tail. So this is the region of your UTR. Are there are two sequences of UTR, two segments of UTR, 5 prime UTR and 3 prime UTR, right? These UTRs are not introns also. These untranslated regions are not introns, but these regions regulate your mRNA localization. It may regulate your stability and protein translation. So these untranslated regions that we are talking about, they are not introns, but these regions will regulate the mRNA localization. It will regulate your stability of that mRNA. How it will regulate the stability? By attachment of RNA binding proteins, and it can also regulate your protein translation by it acts at the initiation level. Okay, the initiation of translation which begins, it regulates at that level by allowing the ribosome to identify that region. So this untranslated region that we are talking now, they are not introns, but these regions regulate the mRNA localization. It will regulate the stability of that mRNA, and also it will regulate the protein translation also. Now the binding of these the binding of RNA binding protein to the 5 prime untranslated region or to the 3 prime untranslated region will increases or decreases the stability of an RNA molecule depending on the specific RNA binding protein that binds. Now which type of RNA binding protein is binding? Which type of RNA binding protein is binding to the 5 prime UTR or to the 3 prime UTR side? Depending on that, it will increase or decrease the stability of an RNA molecule, right? Depending on specific RNA binding protein it binds. So the 5 prime UTR is also called as the leader sequence. You have always heard this term leader sequence in your antibody diversity when you are drawing the heavy chain or when you are drawing the light chain. You have seen such the variable gene segment, the leader sequence followed by a leader sequence, each leader sequence followed by a variable gene segment, then the diversity segment, the joining segment, and the constant regions. So these leader sequence are actually the 5 prime UTR. Okay, the immunoglobulins are pretty much complex. Okay, it has a leader sequence that is your 5 prime UTR, whereas the 3 prime UTR is called as the trailer sequence. Okay, now we will discuss about this 5 prime UTR and 3 prime UTR in detail. First let me talk you the overview here. The 5 prime UTR is upstream. Okay, obviously it is found in upstream. I have shown to you just now the figure. It is found in the upstream from the coding sequence. Within this 5 prime UTR is a sequence that is recognized by the ribosome, which allows the ribosome to bind and initiate the translation process. So the 5 prime UTR is found on the upstream from the coding sequence. It is located upstream outside the open reading frame, and within the 5 prime UTR there are some sequences which will help the ribosome to identify and bind. Now you can able to guess that what will be that sequence. In case of prokaryotes, it is called as the ribosome binding site, the signed Dalgarno sequence, which is found on within this 5 prime UTR. So see, the 5 prime UTR is regulating the translation, right? If there is no signed Dalgarno sequence, how would the ribosome would be able to identify and bind? Now this is occurring in case of prokaryotes, because prokaryotes have this ribosome binding site, right? So within this 5 prime UTR there is a sequence, okay, that is recognized by your ribosome, and which allows the ribosome to bind and initiate your translation process. Whereas the 3 prime UTR plays a critical role in translation termination as well as in the post transcriptional gene expression. So this 3 prime UTR plays a critical role in translation termination as well as post translational post transcriptional gene expression. Okay, let me now talk about the details of this 5 prime UTR. Okay, so coming to this 5 prime UTR now, it is also called as 5 prime untranslated region, also known as the leader sequence. You will also call them as leader RNA. So this 5 prime UTR is a region of an mRNA that is directly found in the upstream from the initiation codon of ORF. The 5 prime untranslated region, also known as leader sequence or leader RNA, it is the region of an mRNA which is directly located upstream from the initiation codon. That is, it is located upstream from the open reading frame. This region is important for regulation of translation of a transcript by different type of mechanisms found in viruses, found in prokaryotes as well as in eukaryotes. So this 5 prime untranslated region is very important for the regulation of translation of a transcript by differing mechanisms in virus, prokaryotes as well as in eukaryotes. While called untranslated, now generally this region is not translated, right? Okay, this region is not translated actually, but sometimes it has been shown that the 5 prime UTR or portion of it, the 5 prime UTR or portion of it sometimes translated into a protein product. This protein product can then regulate the translation of the main coding sequence of the mRNA. So this type of untranslated region also this type of untranslated region, that is the 5 prime UTR, is important for regulation of translation of a transcript by different mechanisms, right, that occurs in virus, prokaryotes as well as in eukaryotes. Sometimes this 5 prime UTR is translated into a protein product. Now this protein product can then regulate the translation of the main coding sequence of mRNA. In many organisms, however, this 5' UTR is completely untranslated, instead forming a complex secondary structure, maybe a hairpin loop-like structure to regulate the translation process. That is found in the virus, the IRES, internal ribosome entry sites. Okay, so this was about the 5' UTR, and I will discussing this now. Okay, so this is the general structure of the 5' UTR of a transcript in eukaryotic organism. So see how the secondary structure has been formed. See the secondary structure that has been observed just before your open reading frame starts. Okay, that is the starting point of your 5' UTR. Now here you can see two AUG codons, right? Two AUG codons you are seeing. Many AUG codons you are seeing here. The UAUG. U for upstream AUG. Upstream start codon. The codons that you can hear you are seeing here. Okay. The codons that you are here you are seeing, where I am trying to draw it. This region. Wait for a minute. Okay, this region. Try to see this region. So this region is not the actual codons. They are the regulatory codons, which are the part of upstream open reading frame. Okay, they are the part of upstream open reading frame which regulates the translation in eukaryotes. Okay, so here you can see the untranslated regions. Okay, the untranslated region at the 5' end. From here your coding sequence starts. You can see here it is written AUG, then your coding sequence has been written in the blue color, in the blue box. That is your main open reading frame. But before that, the start codon and the stop codon that you are seeing here before on the upstream, they are actually your upstream open reading frame, and they are actually a part of your 5' untranslated region. Okay, it can also show the secondary structure which are also regulating. It is also a part of your upstream open reading frame. It is better to say it is also a part of 5' UTR, untranslated region, in the eukaryotic organism. So in many levels this UTR is regulating the gene at the level of transcription, at the level of, especially at the level of translation also. Okay, you have I think I have also discussed about this upstream open reading frame to you in during the translation process. Okay, so this was about the structure of the 5' UTR. Now the 5' UTR has been found to interact with the proteins which are related to metabolism and proteins that translate sequences within the 5' UTR. So this 5' UTR has been found to interact with the proteins which are related to the metabolism, which are related to the proteins that translate the sequence within the 5' UTR. In addition, this region has been involved in transcription regulation, like your sex lethal gene in Drosophila. So this 5' UTR interact with the proteins which are related with the metabolism of the cell, and the proteins which can translate the sequence within the 5' UTR. In addition, this 5' UTR has been involved in transcription regulation. Best example, your sex lethal gene in Drosophila. Regulatory elements within the 5' UTR also has been linked to an mRNA export. The regulatory elements that are found within the 5' UTR has also been used for the mRNA export. Whether the mRNA is now ready for exporting out from the nucleus or not, that can be regulated by your some elements that are found within the 5' UTR. Elements refers to the some sequence, some segments that are found in the 5' UTR. They has been found to regulate with the mRNA export from the nucleus, right, to the cytoplasm. Now the length, general structure, the 5' UTR begins at the transcription start site, okay, and ends one nucleotide before the initiation sequence of the coding region. So the 5' UTR begins at the transcription start site and it ends one nucleotide before your initiation sequence of the coding region. So from the transcription start site, your 5' untranslated region begins and ends just one nucleotide before your initiation sequence of open reading frame. In prokaryotes, the length of 5' UTR is actually 3 to 10 nucleotides long. In prokaryotes, the length of 5' UTR seems to be 3 to 10 nucleotides long, while in eukaryotes it tends to be anywhere from 100 to several thousand nucleotides long. In case of prokaryotic 5' UTR, it is very simple, only 3 to 10 nucleotides long, while eukaryotes, because they will regulate, the prokaryotes also regulate, they have a ribosome binding site or the signed Dalgarno sequence within that 5' UTR. In case of eukaryote, it is much more complicated because they have an upstream open reading frame within this 5' UTR. It can be from 100 to several thousand nucleotides. Okay. Example, STE11 transcript in Schizosaccharomyces pombe. It has 2273 nucleotide within the 5' UTR. So much. While if you want to look at the Lacoperon of E. coli, they have only 7 nucleotides in its 5' UTR. So much difference. Okay, so much difference in the number of nucleotides between the organisms having the 5' UTR. So the different size are likely due to complexity. Obviously, one factor is the complexity of the eukaryotic gene regulation, which the 5' UTR holds, as well as larger pre-initiation complex that must form to begin the translation process. Okay, so main problem is that the complexity of the eukaryotic organisms, they need to regulate, okay, at the level of translation, at the level of transcription, they have to regulate. So that's why eukaryotes have a much larger 5' UTR. Within that 5' UTR, there are upstream open reading frames. Okay, there are some regions which will allow to interact with the ribosomes, maybe, or to regulate the interaction with the ribosomes, while in prokaryotes they have a simple thing, they have a ribosome binding site and other things. Okay, now the elements of eukaryotic and prokaryotic 5' UTR vary greatly. I have already just said the elements that are found within the 5' UTR in prokaryotes and in eukaryotes will be different from each other. In case of prokaryotic 5' UTR, they have a ribosome binding site, also called as signed Dalgarno sequence, and this is the sequence AGGU. This signed Dalgarno sequence will helps to identify the it helps to interact with the 16S rRNA of the small ribosomal subunit, and it is usually 3 to 10 base pairs long, found on the upstream. So in prokaryotes, the 5' UTR have this ribosome binding site, and this is the binding site where your ribosome will interact, because the sequence in the ribosome binding site is complementary with the 16S rRNA of the small ribosomal subunit, which is usually 3 to 10 base pair long. Now if you compare with the eukaryotes don't have ribosome binding site, but they have a Kozak consensus sequence, which increase the efficiency of translation. It says sometimes that Kozak sequence increase the efficiency of translation. In contrast, the eukaryotic 5' UTR have this Kozak sequence, right? A purine nucleotide in the upstream, two nucleotides upstream, and a guanine at immediately downstream from the start codon. Here you can see in the Kozak sequence the AUG is your start codon of the open reading frame. After AUG there is a G, and before AUG in the upstream there are two cytidine has been shown. It can be any nucleotides, it can maybe guanine, it may be adenine, it may be uracil, right? And after these two nucleotides in the upstream there is another A. So presence of, it may be A or G, presence of a purine 2 to 3 nucleotides away from the start codon in the upstream and a guanine at immediate downstream makes your Kozak sequence. So this 5' UTR have this Kozak sequence in eukaryotes, which contains the initiation codon 2. You can see AUG, the initiation codon is also present. The eukaryotic 5' UTR also contains cis acting regulatory elements called upstream open reading frame that I am talking about. I have talked to you about just now. 5' UTR in eukaryotes, they have this Kozak sequence, okay. They also have this upstream open reading frame. Cis acting means found side by side, okay. Trans means from the opposite side it will interact. So these consist of a cis acting regulatory elements found on the same mRNA on the upstream, called upstream open reading frames, and within these upstream open reading frames there is upstream AUG, and there is also a termination codon, just like your natural open reading frames termination codon, which have a great impact for the regulation of translation. I have said that the upstream open reading frame can able to regulate the translation, okay. Unlike prokaryotes, 5' UTR can harbor introns in the eukaryotes. Unlike prokaryotes, these 5' UTR can harbor introns in eukaryotes. They can be recognized as introns, but they are not introns. In human, 35%, approximately 35% of all genes harbor introns within the 5' UTR. This is not little essential for you to see for about the regulation. You have to see this much, you have to study this much here, that what is the difference of the 5' UTR among the prokaryotes and eukaryotes. Prokaryotes have a ribosome binding site, which is also called signed Dalgarno sequence within this 5' UTR, which can able to interact with the 16S rRNA, and there are 3 to 10 base pairs upstream. uh, it also have in case of eukaryotes they have a Kozak sequence. Kozak sequence are found within the 5' UTR, including your initiation codon, the main codon of the open reading frame. The eukaryotes also have uh, what can I say, the upstream open reading frame, the UORF, within this 5' UTR. Within this 5' UTR in eukaryotes, within this 5' UTR there is an upstream open reading frame which will able to regulate the translation, because upstream open reading frame have a upstream AUG. They have also a stop codon that can helps to regulate the translation. Okay, now next one. Now as this 5' UTR have a high GC content, the 5' UTR have a high GC content, that's why secondary structures often occur within it, like your hairpin loops, as I have shown you in the figure little bit ago. Hairpin loops formed, and they are one of the most secondary structures that can be located within this 5' UTR. This secondary structure can also affect the regulation of your translation process. Okay, so 5' UTR, due to its high GC content, secondary structures like hairpin loop form, and this hairpin loop can regulate that regulation of translation. Okay, it cannot able to regulate the transcription because it is located in the 5' upstream. The regulation of translation in eukaryotes is more complex than prokaryotes. Obviously, regulation of translation in eukaryotes is more complex than prokaryotes. Now, initially, if you remember about the initiation of transcription in eukaryotes, the EIF4F complex is recruited to the 5' cap. Just remember and think about the transcription initiation in eukaryotes, the 48S in pre-initiation complex. Remember, on one side your ribosome getting the small ribosomal subunit getting assembled with the initiation factors and recruits the tRNA to the P site of the ribosome, whereas on the other side the EIF4F complex interacts with the 5' cap, right? So the EIF4 complex in eukaryotes is recruited to the 5' cap, which in turn recruits the ribosomal complex to the 5' upstream untranslated region. The EIF4F complex recruited to the 5' cap, which in turn it recruits the ribosomal complex to the 5' untranslated region. Both EIF4E and EIF4G binds to the 5' UTR, which limits the rate at which the translation initiation can occur. So both EIF4E, which is a type of initiation factor, and EIF4G, they bind to the 5' untranslated region, which will limit the rate at which translation initiation can occur. However, this is not only the regulatory step of translation that involves 5' UTR. So, regulation of translation of eukaryotes is complicated. Initially, your EIF4F complex interacts with the 5' cap and recruited to the 5' cap, which in turn it recruits the ribosomal complex to bind to the 5' UTR. Then it will scan the DNA to it will scan the RNA to identify the start codon. Both EIF4E and EIF4G binds to the 5' UTR, which will limit the rate at which translation initiation can occur. However, this is not only the regulatory step of translation that involves 5' UTR. RNA binding protein sometimes serves to prevent this pre-initiation complex from forming. There are some RNA binding proteins which serve to prevent the pre-initiation complex from forming. It prevents the formation of that 48S pre-initiation complex, right? There are some proteins which prevents that formation of 48S pre-initiation complex. An example is regulation of the MSL2 gene. The protein SXL attached to the intron segment located within the 5' UTR segment of the primary transcript, which leads to inclusion of the intron after processing. So sometimes RNA binding proteins prevent this pre-initiation complex. It prevents this 43-8S pre-initiation complex from forming. Best example is regulation of your MSL2 gene. How the MSL2 gene is regulated? There is a protein called SXL which attached to the intron segment located within the 5' UTR segment of your primary transcript. So there is a protein called SXL. It binds to the 5' UTR, which leads to inclusion of intron after processing. This sequence allows the recruitment of proteins that bind simultaneously to both the 5' and 3' UTR, not allowing translation proteins to assemble. However, it has also been noted that what happened, the 5' the SXL protein and RNA binding protein bind to the 5' UTR, right, and led to inclusion of the intron after processing. And this sequence will allow recruitment of those proteins that bind simultaneously to both 5' and 3' UTR, not allowing the translation process to assemble. It forms a circle of mRNA, right? The 5' UTR found on the upstream, the 3' UTR found on the downstream after the open reading frame. So they will bind together, interact together and form a circle. I will show you the figure how the circle form. Maybe like here, like this. You can see here interaction between the proteins bound to the 3' UTR and 5' UTR causes circularization that regulates the translation. Okay, so in this way. So this protein is SXL. You are talking about the example that how RNA binding proteins they prevent the pre-initiation complex. They prevent that 48S pre-initiation complex from forming. The protein, RNA binding protein called SXL, they bind to the 5' UTR segment, which leads to the inclusion of the intron after the mRNA processing. This sequence allows the recruitment of the proteins that will bind simultaneously to the 5' UTR as well as to the 3' UTR, not allowing the translation process to assemble also. However, it has also been noted that SXL can also repress the translation of RNA that do not contain poly A tail or more generally the 3' UTR. Another important regulator of translation is your interaction between 3' UTR and 5' UTR. So you can see here 3' UTR and 5' UTR getting interacted to form circularization of mRNA that can regulate your translation. So this closed loop like structure will inhibit the translation. This has been observed in Xenopas laevis. Okay, this has been observed in Xenopas laevis. Xenopas laevis is what? A type of frog, right, in which your EIF4E bind to the 5' cap, interacts with the masking bound to CPEB on the 3' UTR, creating a translationally inactive transcripts. This translational inhibition is lifted once CPEB is phosphorylated, displacing the masculine binding site, allowing for the polymerization of the poly A tail, which can recruit the translational machinery by means of PABP. So, however, it is important to note that this mechanism has been a great scrutiny. So this is the way that which your translation regulation can be done by your 5' UTR and 3' UTR by circularization of the mRNA, okay, which will inhibit the translation process. Okay, so you will just say the overview, not in details, about this circularization, that it may also inhibit your translation process. Now, another form of translational regulation is your UORF, the upstream open reading frame. They are found 35 to 49% of all human genes. An upstream open reading frame is located on the 5' UTR, located in the upstream of the coding region of initiation site. These upstream open reading frame have their own start codon and stop codon, and their start codon is also AUG, also known as UAUG. This codon can be scanned by ribosomes and then translated to create a product, which can regulate the translation of the main open reading frame. So this upstream open reading frame will be translated by your ribosome to form a protein product, and that protein will regulate your main open reading frame. The translation of the protein within the main open reading frame after a UORF sequence has been translated is known as reinitiation. After your UORF, there is a ORF, the main open reading frame. So that region again you are getting initiated, that is called as reinitiation. So the translation of the protein within the main open reading frame, just after the UORF there is a main open reading frame, that point is known as reinitiation. So your reinitiation will be, this is a 5' UTR. Here it has not been shown that upstream open reading frame, but after the upstream open reading frame then comes your main open reading frame, the coding sequence, where the main start codon will starts. That is called reinitiation. Okay, that is called your reinitiation. So the process of reinitiation is known to reduce the translation of the ORF protein. So because upstream open reading frame is allowing the translation to begin, then after so much of translation process, the ribosome will leave off from that mRNA and not doing the translation of the main open reading frame because of utilization of the amino acids for the formation of protein because of that upstream open reading frame. So due to this reason, the ribosome will leave off the complex and it will not be able to translate the main open reading frame. In this way, it can also the upstream open reading frame found on the 5' UTR can also regulate the translation. Now in case of virus have a site within the virus also have a 5' UTR known as internal ribosome entry sites, or IRES, which is a cap independent method. Virus don't have a 5' cap, but they have a internal ribosome entry sites found within the 5' UTR, which is a cap independent method of translational activation. Instead of building a complex at the 5' cap, these internal ribosome entry sites called IRES allow for direct binding of the ribosomal complex to the transcript to begin the translation process. The IRES enables the viral transcript to translate more efficiently due to lack of need of pre-initiation complex, allowing the virus to replicate quickly. So this IRES found within the 5' UTR, it enables the viral transcript to translate more efficiently due to lack of needing a pre-initiation complex, allowing the virus to replicate more quickly. So this was all about the 5' UTR. I will discuss this 3' UTR in the next video.